So basically, once these pups grew up and became older, they then went on their first date. This Nova1 gene is definitely unique to humans. Neanderthals and Denisovans did not have the same gene either. It might not be just a language gene, it might be a gene responsible for a lot of other things that make us human.
For centuries, they have been searching for that one determining factor that causes humans to be so different. A search that is focused in exaggerated fashion on the now extinct Neanderthal. All this in an effort to show that intelligence really started in the Eurasian continent. But recent genetic studies are not only dismantling the notion of a cognitively potent Neanderthal, but it is at the same time upending the distorted notions that those who have been blessed with melanin haven't received those key genes. And yet, what is emerging is that it is well within the continent of Africa that the human sparks started before spreading to the rest of the world, and that in fact, those hominids outside Africa did not have the key elements that make us special. And every discovery, as we shall see, only confirms that fact. Skull size measurement, melanin concentration, IQ tests, clearly proven not to measure intelligence, but to measure Western cultural affinity instead. From Count of Gobineau to Samuel George Morton, many things have been tried to deny the black man and the black woman their humanity. What started out as a means to morally justify the human trafficking that occurred across the Atlantic has now taken a life of its own. A series of genes, the TKTL1 gene, the FOXP2 gene, and the NOVA1 gene, are all showing that the key elements of human cognitive development emerged in Africa and were present in all Africans before humans went out of the continent to displace other hominids. One groundbreaking finding came in 2013 with the discovery of Homo naledi in South Africa. Paleoanthropologist Lee Berger's team uncovered over 1,500 fossils from at least 15 individuals in the Rising Star cave system. Homo naledi shocked scientists because it exhibited advanced behaviors despite having a very small brain. Notably, naledi deliberately buried its dead, a practice suggesting symbolic thought and self-awareness. This behavior implies a concept of the self and, perhaps, an understanding of life and death, hallmarks of higher intelligence. The small-brained naledi also likely made and used tools, indicating that brain organization and neural capacity mattered more than sheer size. These findings reinforce a crucial point: brain size alone does not determine intelligence. In fact, one of history's greatest minds, Albert Einstein, had a brain weighing only 1,230 grams, smaller than the average adult male's 1,400 grams. Yet certain regions of Einstein's brain, related to mathematical and spatial reasoning, were unusually well developed. This highlights that it is brain structure and connectivity, not mass, that underpin genius. If we judged by cranial capacity alone, we might falsely label Einstein or Homo naledi as unintelligent, and yet. Modern studies confirm that neural architecture and metabolic efficiency are far more important for cognitive performance than brain volume. For example, Richard Heyer et al. Found that brain metabolic rate, how much energy the brain consumes, correlates with cognitive performance better than overall brain size. The more neurons and synapses a brain has, and the more energy it uses, the more computational power it has. Likewise, another study showed that intelligence relates to the complexity of brain connectivity rather than cranial dimensions. Crucially, when researchers compared Neanderthal brains to modern human brains, they found stark differences in function despite similar size. Neanderthals likely had a lower cerebral metabolic rate and distinct mitochondrial function, hinting that their brains operated less efficiently. In other words, Neanderthals had the hardware, large brains, but seemingly not the same software, energy usage, and neural wiring as Homo sapiens. This dispels the romanticized notion that Neanderthals were just like us. Evidence increasingly shows they were not on par with modern humans in cognitive abilities. So, what does that mean? That means that when you hear of people bringing forth the argument that one ethnicity in modern humans are more intelligent because they have a bigger brain, you should completely reject these notions. In fact, we have known this to be true for a long time, because women have, on average, a smaller brain size than men. And yet, today, now that they have been given a chance, women significantly outperform men in all levels of academic field. But the TKTL1 gene did more than change the architecture of ancient human brains. It had a mutation that explains a lot in our current condition. If brain size isn't the key, what is? Recent genetics research points to specific mutations that turbocharged human brain development. A single mutation in TKTL1 set our ancestors on a different cognitive path. TKTL1, transketolase-like 1, plays a crucial role in the developing brain's production of neurons. Modern humans have a unique version of this gene that Neanderthals and apes lacked. In a 2022 study, scientists found that the modern human variant of TKTL1 differs by just one amino acid from the archaic version. Yet this tiny change had an enormous impact. When researchers introduced the human TKTL1 gene into developing mouse brains or organoids, it quadrupled the production of neuron-generating cells, basal radial glia, in the frontal cortex. As a result, the mice grew many more neurons than those with the Neanderthal-type gene. In contrast, the Neanderthal-like TKTL1 did not boost neuron numbers. The study's authors conclude, modern humans differ from apes and Neanderthals by this single amino acid change. The human variant of TKTL1 drove more generation of neuroprogenitors than did the archaic variant. The authors suggest that the modern human has more neocortex to work with than the ancient Neanderthal did. More neurons, especially in the frontal lobe, mean a greater capacity for complex thought. Each neuron can form thousands of connections, synapses, with other neurons, so increasing neuron count explosively multiplies the potential synaptic network. This network density is what enables abstract reasoning, problem-solving, and creativity. When the TKTL1 mutation swept through our African ancestors, it likely triggered a non-linear leap in cognitive capacity. More frontal neurons meant the brain could develop more intricate circuits for planning, social cognition, and other high-level functions. Importantly, Neanderthals did not have this mutation. But intriguingly, evidence suggests Homo naledi did possess the modern TKTL1 gene. Despite Naledi's small brain, this gene may have helped it achieve human-like intelligence, further proof that brain wiring can trump brain size. In sum, the TKTL1 gene discovery reveals that the foundation for a smarter brain was laid in Africa. This beneficial mutation arose in an African population of early Homo sapiens roughly 300 to 400,000 years ago and gave them a neurological edge. Those humans, with their expanded neocortex, would eventually spread out of Africa and replace other hominids. As we will see, another genetic change, one affecting how we communicate, was the next critical piece of the puzzle. Another genetic innovation unique to modern humans is a variant of the NOVA1 gene. NOVA1, neuro-oncological ventral antigen 1, encodes a neuron-specific RNA-binding protein that is crucial for brain development and neural function. It helps orchestrate how neurons form connections and networks, partly by regulating alternative splicing of other genes during brain development. NOVA1 is so vital that disrupting it can cause severe neurological problems. In fact, laboratory mice cannot survive without some functional NOVA1, and in humans, certain NOVA1 mutations lead to developmental disorders. What makes Nova 1 especially interesting is a single amino acid substitution that appears only in modern humans. Virtually all other mammals, and even birds, have an identical Nova 1 protein. But humans carry a unique change. At position 197 of the protein, we have a valine, V, instead of isoleucine, I. This Nova 1 I197V variant is absent in Neanderthals and Denisovans, our closest extinct relatives. Geneticists confirmed this by comparing genomes. Neanderthal and Denisovan DNA encode the archaic Nova 1 sequence, isoleucine at 197, whereas every sequenced Homo sapiens, barring a handful of rare exceptions, has the valine substitution. In an analysis of over 650,000 human genomes worldwide, all but 6 carried the modern Nova 1 gene. Those 6 individuals had the archaic version, likely due to ancient interbreeding with archaic humans. This implies that the human Nova 1 mutation arose in our African ancestors after the split from Neanderthals, 250 to 300,000 years ago, and swept rapidly through the population. As one researcher put it, our data show that an ancestral population of modern humans in Africa evolved the human variant, which then became dominant. This population then left Africa and spread across the world. Why would a single genetic tweak become nearly universal in our species? Recent experiments suggest Nova 1 helped spark the evolution of complex language and communication. Dr. Robert Darnell and colleagues at Rockefeller University used CRISPR gene editing to create humanized mice, replacing the normal mouse Nova 1 gene with the human I197V variant. They then observed how this affected the mice's brains and behavior. The results were astonishing. Mice with the human Nova 1 began speaking differently to each other. Baby mice typically emit ultrasonic squeaks to call their mothers, categorized into a few basic sound types, often labeled S, D, U, and M. Mice carrying the human Nova 1 variant produced calls with a different mix of these sound letters, effectively using a shifted vocabulary. Their calls were higher pitched and included sounds normal mice don't usually make. As they grew, the changes became even more pronounced. Adult male mice with human Nova 1 made more complex courtship songs when interacting with females compared to control males. They talked differently to the female mice, Darnell explains. The modified male's wooing calls had richer patterns and higher frequencies, indicating a greater vocal range and perhaps more elaborate communication during mating. Overall, researchers noted that the mice with human Nova 1 squeaked in more intricate sequences than their unaltered counterparts. Even the rhythm and structure of their sounds changed. Some of the ultrasonic letters that make up mouse calls were replaced with new ones. These vocalization experiments suggest that Nova 1 affects the neural circuits of communication. Indeed, molecular analysis showed that the human Nova 1 protein in mice bound to and altered the splicing of a suite of genes linked to vocalization and neuromuscular control of speech. Incredibly, the only major differences detected in the mice's brains were in RNA processing for genes related to making sounds. Other aspects of brain development appeared normal. It's as if Nova 1's human variant is finely tuned for the wiring of speech and language networks without disturbing general brain functions. Many of these vocalization-related genes were found to be binding targets of Nova 1, further suggesting the involvement of Nova 1 in vocalization, said Dr. Yoko Tajima, the study's first author. Equally fascinating were the social behaviors of the humanized mice. Researchers observed that modified mice engaged in longer conversations with each other, spending more time exchanging calls than typical mice. The males' enhanced songs appeared to get responses from females, hinting at a feedback loop of communication. And only mice with the humanized version of Nova 1 would mate with each other. They did not want to mate with mice that had the archaic variant. This is interesting, as it points to a natural selection based on higher communication abilities. In another study, scientists took the archaic version of the Nova 1 that is present in Neanderthals and introduced it to human cells. And I quote, "Both Neanderthals and Denisovans had one version, and nearly all modern humans had another, suggesting positive selection." Insertion of Neanderthal gene variant of the neuro-oncological ventral antigen 1, NOVA1 gene into human cortical organoids might promote slower development and higher surface complexity in the brain models. In evolutionary terms, if a new gene variant helped early humans communicate more effectively, for example, to woo mates or coordinate group activities, those with the gene would have a selective advantage. One can imagine how such changes in vocalization could have a profound impact on evolution, Darnell notes. Improved communication could strengthen social bonds, improve cooperation, and ultimately enhance survival. Not surprisingly, NOVA1's importance is also evident in human patients. Mutations in NOVA1 lead to speech and learning disabilities. Darnell's team has identified cases where changes in NOVA1 are associated with developmental language and motor delays. In one recent report, a child with only one functional copy of NOVA1, a condition called haploinsufficiency, exhibited neurological symptoms, including significant speech delay. NOVA1 is also among the genes linked to autism spectrum disorders, particularly those affecting nonverbal communication. These observations underscore that NOVA1 is essential for normal human communication and cognitive development. It likely helped structure our ancestors' brains for abstract thought and spoken language, complementing the raw computational boost provided by TKTL1. In essence, TKTL1 gave us a bigger engine, more neurons, while NOVA1 fine-tuned that engine for the complex song of human language. No discussion of a speech gene can ignore FOXP2, the first gene ever linked to language. Discovered in the early 2000s, FOXP2 was hailed as the genetic key to human speech. People with mutations in FOXP2 have severe speech and language difficulties, struggling to coordinate the mouth and tongue movements required for speech. Researchers found that humans have two unique amino acids in FOXP, two not present in other primates, suggesting these changes might have been crucial in our lineage. However, subsequent discoveries complicated this story. It turned out Neanderthals also carried those same FOXP2 mutations, meaning the variant predates the split between Neanderthals and modern humans. If FOXP2 alone made the difference, Neanderthals should have had speech capacities similar to ours. Yet the archaeological and anatomical evidence for Neanderthal language is ambiguous at best. In 2018, a comprehensive analysis of diverse human genomes found no signs of recent natural selection on FOXP2, undermining the idea that it drove a late, dramatic leap in language. As one expert noted, FOXP2 was effectively deposed as the singular language gene. This opened the door for other candidates like NOVA1 to take center stage in explaining why only modern humans have fully developed language. Experiments inserting human FOXP2 into mice have yielded intriguing results, and they differ from the NOVA1 findings. Mice with humanized FOXP2 do not suddenly start chatting more complexly. In fact, one study found their ultrasonic vocalizations were not significantly changed. What did change was how the mice learned. In a 2014 study, mice engineered with the human FOXP2 variant learned to run a maze much more quickly than normal mice. They were better at converting new experiences into habitual routines. In other words, FOXP2 made the mice more efficient at a certain kind of learning, procedural learning. The FOXP2-enhanced mice could take a task that initially required conscious effort and perform it more automatically, operating on autopilot based on cues, as the researchers described. This supports the idea that FOXP2 might help with the automation of motor sequences. Speech is, after all, a complex motor sequence we perform effortlessly. However, this boost in habit learning came with a potential trade-off. Observers noted that the humanized FOXP2 mice became less exploratory than usual, seemingly content to stick with learned routines. They also exhibited altered brain chemistry. Measurements showed differences in dopamine levels in the striatum, a brain region critical for learning and motivation. In fact, one part of the striatum, the dorsomedial section, linked to goal-directed behaviors and exploration, had dopamine levels drop to about 70% of normal in FOXP2 humanized mice. This suggests that while FOXP2 may help ingrain patterns, which is useful for language fluency, it might also dampen novelty-seeking or flexibility. High dopamine typically drives curiosity and reward-seeking. A reduction in certain circuits could make an animal less inclined to explore new possibilities. Thus, instead of enhancing general intelligence, the human FOXP2 made mice more routine-driven. As science writer Ed Yong quipped, the mice were less exploratory than usual, despite having the so-called language gene. This indicates that FOXP2's evolutionary role might have been more about refining speech motor control and efficiency rather than sparking creative intelligence. It also aligns with the fact that FOXP2 is not unique to modern humans. Neanderthals had it, and it alone didn't save them from extinction or grant them civilization. The contrast between Nova1 and FOXP2 is illuminating. FOXP2, once hyped as the speech gene, is now seen as just one piece of a larger puzzle, and not a particularly differentiating one at that, since archaic humans had it. Nova1, on the other hand, truly sets modern humans apart. The human Nova1 variant appears to have been part of a sweeping evolutionary change that gave our species a communication advantage. As Dr. Darnell remarked, Nova1 may be a bona fide human language gene, though certainly it's only one of many human-specific genetic changes. It likely worked in concert with mutations like TKTL1 and others yet to be discovered to empower the modern human brain. Taken together, these genetic discoveries highlight an important truth. The most critical advancements that made us truly human arose in Africa, long before humans migrated across the globe. The TKTL1 mutation originated in Africa around 300,000 to 400,000 years ago, giving rise to bigger neural networks in the frontal cortex of early Homo sapiens. The Nova1 mutation, I197V, likewise appears to have emerged in an ancestral African population roughly 250,000 to 300,000 years ago. It spread rapidly because it conferred a vocal communication advantage, allowing richer expression of ideas and coordination within groups. Those enhanced humans, our direct ancestors, then left Africa and, with their superior brains and communication, outcompeted other hominids like Neanderthals and Denisovans. This means that all modern people, whether of African, European, Asian, or other descent, owe their brains' prowess to the same ancient African innovations. Early African Homo sapiens evolved the hardware, more neurons, and software, advanced language ability, that would later spark art, science, and civilization. When these humans dispersed out of Africa, they carried those gifts to every corner of the world. As geneticist Svante Pääbo's work showed, only a handful of genes separate us from archaic humans. It's not about having bigger brains, but about better brains. Brains wired for creativity, adaptability, and shared knowledge. And the evidence points to Africa as the cradle of those defining traits. In fact, the two most ancient and influential civilizations, those of the Nile Valley, ancient Nubia and Egypt, and Mesopotamia, Sumer, were established by descendants of these early innovative humans, and significantly, by Black African peoples in their early stages. They pioneered writing, mathematics, medicine, architecture, and more, long before others. This historical reality was long obscured by dishonest and ill-motivated scholarship, but it aligns perfectly with the genetic evidence. The spark of intelligence was lit in Africa. Africans not only developed the biological capacity for high intelligence, they were the first to ignite the flames of civilization. They then shared those innovations with the rest of humanity as migrations and cultural diffusion occurred. Those who have been following the channel for a while know that I do not subscribe to any notion of racial superiority. Neither Caucasian, Levantine, Oriental, nor black superiority. And this is simply because all humans display the same cognitive ability and the same cognitive potential. And the more scientists look, the more they find that we are the same. In fact, those elements that cause observable differences in humans are those to which we need to adapt the quickest. Environmental factors. In only a few generations, those who have too much melanin would lose their melanin if they live too far from the equator, and vice versa, those with too little melanin would get out-competed by those who are adapted to high solar exposure. Physical traits, like skin pigmentation, body size, or lactose tolerance, are often under strong selective pressure from environmental factors such as climate, diet, or disease. These traits can show relatively rapid changes across generations when the pressure is intense. For example, skin pigmentation is a classic case. Populations migrating to regions with less sunlight, like Northern Europe, developed lighter skin over thousands of years to optimize vitamin D synthesis. Genetic studies, such as those on the SLC24A5 gene, suggest this adaptation occurred within 5,000 to 10,000 years in some groups, which is fast in evolutionary terms. Studies like those from the Human Genome Project or population genetics research, e.g. by David Reich's lab, show that adaptations to local environments often prioritize physical traits, first because they're more straightforward responses to tangible pressures. For instance, Tibetan high-altitude adaptations, EPAS1 gene, for oxygen efficiency emerged within 3,000 years, purely physical. Meanwhile, intelligence-related genes are harder to pin down. Twin studies in GWAS, genome-wide association studies, suggest cognitive traits evolve under weaker, more gradual selection. All humans today possess the genetic endowment for high intelligence, and no group is inherently more evolved in this respect than another. The very concept of distinct biological races ranking intelligence is a myth with no basis in genetics or history. Instead, what the research shows is a unifying story. Our species' intelligence is a legacy we all inherit from a common African past. The NOVA1 and TKTL1 genes, among others, are part of a genetic toolset that made abstract thought, complex language, and culture possible. These were universal gifts, not the province of any one race. One surprising thing is that Dr. Cheikh Anta Diop has predicted all of this 50 years ago in his studies, well ahead of genetic studies, and he was spot on. If you want to support Without History, consider becoming a Patreon or joining our YouTube membership. I would like to leave you with an homage to a few members of the community that are very hard at work to educate the masses about true Black and African contribution to history.
It's Big B, the prolific producer.
Mr. Imhotep. May he rest in peace. I posthumously grant him the title of Dr. Imhotep for his service to humanity. The Debunker, a reserved, silent, but potent researcher that helps many of us behind the scenes with his amazing insights. Joe Bassi, the typical African. I don't know how he can post so much on social media. A must follow. Zach Mwakasa. He speaks the truth, plain and simple. But behind his imposing beard, he is seeking to liberate many of us from the mental sickness we suffer. You know I have to mention Kamjiverse. Always under-celebrated, he is a wealth of knowledge and gems you cannot miss.
It's Big VDV living prolific.
King Cam, a cultural and literal teacher who we cannot do without. Remember, many have been raised to believe that Africa and black people are without history. You are now part of the army to let them know that those who are said to be without history birthed civilization itself.